Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you not one, but two really awesome guests today involved in science and technology that's creating a better tomorrow. Today, we have the honor, first, of being joined by Dr. Jack Hoppin, who is a chairman and chief executive officer of Radiotherapeutics, which is a discovery through early clinical phase pharmaceutical company that's employing a really interesting and innovative suite of technologies to develop best-in-class targeted radiotherapeutics for treatment of a range of cancers. Dr. Hoppin holds a Ph.D. in applied mathematics, University of Arizona. He also worked as a Alexander von Humboldt postdoc fellow at the Research Center Jülich, Germany. Previously, Dr. Hoppin was the president of Konica Minolta Precision Medicine, co-founder and CEO of Invicro, vice president of imaging systems at Bioscan, and has a couple decades of experience in imaging research and has been a member of the World Molecular Imaging Society Board of Trustees. Also have the honor of being joined by Dr. John Babich. He's the director, president, and chief scientific officer of Ratio, has been an active researcher in the field of molecular imaging and targeted radionuclide therapy for over 3 decades. Previously worked at the Weill Cornell Medical College for several years as Chief of Radiopharmaceutical Sciences, was president and chief scientific officer at Molecular Insight Pharmaceuticals, a biopharmaceutical company focused on the discovery and development of these innovative radiopharmaceuticals. He also served as Assistant Professor of Radiology at Harvard Medical School, as well as Principal Nuclear Pharmacist at Mass Gen. Dr. Babich has a Ph.D. with a focus on biophysics, medical physics, and radiation biology from University of London, a master's in radiopharmacy from USC, and did his undergraduate in pharmaceutical sciences at St. John's University. Honored to have them both with us. A lot of really cool topics to get into today. Dr. Jack Hoppin, Dr. John Babich, thank you so much for taking time to come on the show.
Thanks for having us. Great to meet you.
Yeah, great having both of you. Really exciting topic that I'm looking to get into. Would love to start things off, though, as we typically do, by handing both of you the floor for a couple minutes, just to further introduce yourselves. I love taking a look at our guest background, and Jack, I noticed that you did your PhD back in the day in medical imaging on this interesting regression without truth model, when we don't have a gold standard for something, but would love to hear the background of how you got involved in this space, because obviously imaging, and we'll get to John in a minute, but obviously major connection to all things radionuclides, and love to hear a little bit more of where you started out.
Oh, great. Well, thanks, and thanks for having us. Yes, yeah, my PhD thesis was actually on an applied statistics problem that was born out of an imaging conversation, but the lab I worked in was the lab of Harry Barrett at the University of Arizona. It was the Center for Gamma Ray Imaging in optics and radiology, and there worked on single SPECT and PET imaging, which are common clinical imaging modalities, and then did a postdoc at a German national lab, as you mentioned, working on the development of a micro SPECT CT. So SPECT is a ubiquitous tool in nuclear medicine. It's an old imaging technique. It's mostly been replaced by PET, which is far superior clinically. But at that lab, I worked in the lab of Neil Schramm. We worked on making a SPECT imager for rodents, and then after my postdoc, which sounds like a strange activity, but it was very useful in radiolabeling a drug and imaging its distribution over time. And so after my postdoc, an American company had in-licensed that, Bioscan, you referred to them. And I moved to Hungary, where we worked with a company called Mediso to manufacture that product. So I was on a track to being a college math professor, and John and I both talked about taking right-hand turns. I took a right-hand turn into industry. Was not obvious I moved to Budapest after that postdoc, but and that product was successful. Got to really see the landscape of people doing radiolabeled drug research, both in pharma, in industry, and in academia. Became very enamored with how productive pharmaceutical companies were at using imaging for discovery as well as readouts and trials, and started a company in 2008, the week of the financial crisis, conveniently, to do imaging in drug discovery and later in development. And so have been studying radiolabeled drugs for a long time, and their kinetics and their properties, which really lends itself to what we do and what we're going to talk about today. And that company was acquired, and I stayed on in that company for a few years out of passion, and it was a great experience working not only on that, but also Konica Minolta had acquired Ambry Genetics, a large-scale hereditary genetic testing company, and had a digital pathology business unit. So it was a great experience. And at that time, and I'm going to hand this over, we were seeing a lot of ideas in this class of drug we're going to talk about, which is not a new idea, but was really coming, for reasons we'll cover today, coming to the forefront. And I certainly imaging, the evolution of imaging, the evolution of this class of drug, all these technologies kind of are synergistic. And we were seeing a lot of ideas coming to our company to study them, to do contract research for them. And John knocked on. We met. We were familiar with each other from the field, and John has been a pioneer in this space. Has a great reputation and a great thinker. And I was on the call. I don't like to compliment him in front of his face, but I did hear, and it's fine. And then John, we connected in 2018, while John was at Cornell, and he had what I thought was the right idea. And John, with that, why don't you give your background and get us up to?
Yeah, thanks, Dan. It's a great introduction, and Ira, thanks for having us.
Sure.
Yeah, so my background, you mentioned it. I think you did a pretty good job, probably better than I would have. But I'll just go back to my time in the UK. So when I was at the Institute of Cancer Research and the Royal Marsden Hospital, I really got exposed for the first time to targeted radiotherapeutics, and that was particularly in the space of treating children with neuroblastoma.
Right.
There was a big push at the time, and it wasn't even new then. I guess that was back in '90, but there was a lot of work that came out of University of Michigan. There was a lot of pioneering people there that back early in the nuclear medicine field had come up with many things. Incredible capacity for ingenuity there, and they had developed a drug called MIBG. And it's taken up in tumors that have what's known as uptake 1 systems. So these are sort of sympathetic adrenergic type tissues. So tumors such as pheochromocytoma, paraganglioma, and neuroblastoma all seem to have this neural crest phenomenon. And you can take the drug acts as a carrier, so it'll get pulled into these tumors. So I worked in that space in clinically with children, with our medical oncology colleagues, but also was simultaneously doing a lot of work with, as you can imagine, antibodies were the big thing at the time. They were kind of new and exciting back in 1985, and it wasn't that far after they were discovered as monoclonals that lots of people were jumping in and looking at that. And we did a lot of animal work, and that animal work was exciting because you could target tumors in animals quite nicely. Took several of those antibodies into the clinic and really never saw the translation occur in any significant way. So whatever was promising in the mouse somehow is not translating in humans. Unlike this small molecule called MIBG, which was, I would say, modest in the mouse, but very dramatic in children and people and adults with these types of cancer. So started to think about at that point, and this will bear some fruit later on, of molecular size being an important part of the design and understanding of targeted therapeutics. Fast forward there, I went from the ICR and the RMH to MGH, and I was at MGH for 8 years and doing a lot of, as Jack was saying, we were doing a lot of radio labeling of drugs for pharma at MGH. So, I was on an initial contract there, Pfizer, for labeling fluconazole, the antifungal, because at a lot of time, at that point in time, the question was, will it penetrate the blood-brain barrier? Can you actually treat people with CNS fungal infections? It was a very important indication, and we showed that that was possible. Lots of other drugs in the meantime, and was looking at ways of actually, how can we think about making targeted radiotherapeutics that showed promise in mice as antibodies, but weren't translating due to some structural perturbation or structural feature. And so we started to think about peptide mimetics, smaller molecules, and so on and so forth. So fast forward, spend a lot of time thinking about that. It's a lot of time trying to think about, how do we make these targeting agents better, pharmacologically, structurally. And then I had an opportunity to start a company with a couple of other colleagues at MGH in Boston, and we started Molecular Insight Pharmaceuticals, and that sort of kind of broke open in many ways, what we're doing at Ratio today. Not so much that it's the same idea, but we jumped into a space that right at the time was really about molecular imaging, but simultaneously was showing its promise in the possibility of therapy. Right. So that these small molecules, while they could be labeled with a positron emitter for PET imaging, as Jack alluded to, is a very highly sensitive and quantitative imaging device for imaging and radiological assessments of disease and so on and so forth. Could you take some of those molecules and change the isotope on them, whether you put lutetium or actinium, and put them on those molecules for therapy? And that was just starting to happen. We came up with some very nice molecules for imaging prostate cancer. That turned into, these were the PSMA molecules, that turned into a opportunity to do therapy with those same molecules. And we started with iodine-131. Those were the first therapy molecules that went into people in the world, with a colleague over in Heidelberg by the name of Professor Haberkorn. And that really cracked open the PSMA world for therapeutic opportunities. And that really was a tremendous opportunity for the field and for patients with prostate cancer globally. And so in that process, was this chief scientific officer there for 15 years. That company got sold to Progenics. Progenics ultimately got acquired by Lantheus, just pull that out there. But in the meantime, I had the opportunity to, either stay with Progenics, which I didn't want to do, and because I had this opportunity came my way, would you like to come set up a lab at Cornell Medical College in New York City, which was highly attractive, and lots of thinking, lots of great people, lots of opportunity, and they had embraced targeted radiotherapy there in a big way. Scott Tagawa, who's a medical oncologist and the GU, heads up the GU section there, had already embraced that with a fellow by the name of Neil Bander. And so there was a real dramatic interest, I would say dramatic relative to lots of other places I could have gone. And so hence I went there for 8 years. And when I was there, to get to coming back to the arc is long, but it'll get there. One of the things we realized is that pharmacokinetics had a lot to do with how well these drugs were behaving. So if you think about targeted radiotherapeutics, kind of analogous to real estate, right? Location. So it's really about there isn't a pharmacological component to this, except in the delivery and the attachment of the molecule to the cell, right? So we're not trying to do something downstream, like we don't have a BRAF inhibitor, we don't have a kinase inhibitor, right? It stops other things from happening. What we have is the ability to, and we can potentially make use of those interactions, but we really want this to be a sort of a Trojan horse, that the molecule is attracted or is recognized by something on the cancer cell, where it can bind and stay there for sufficient amount of time to deposit the energy that's locked in that isotope. And what we're seeing is that the pharmacokinetics really, because we had taken lots of compounds into people in Europe, the pharmacokinetics really were driving a lot of what was happening there. Not just the affinity, not just the the specificity for the target, but this other component piece. And so we thought, no one really thinks about designing pharmacokinetics into a molecule. They think about, how selective is it and how potent is it?
Right.
Turns out with radiopharmaceuticals, because it's important where it goes, but it's also important where it doesn't go, right?
Yep.
Because if it went everywhere to the same amount, even if you got a lot in the tumor, you'd have no therapeutic index, right? You'd have no therapeutic ratio. It'd be 1. So everything gets irradiated the same way. And just like anything else in targeted in external beam radiotherapy, you have to make sure that you can hit the tumor selectively, or at least normal tissue to a much less extent, in order to not create damage in the surrounding tissues. So how do you do that with an injectable drug? And one way to do that is to sort of put a guidance component into the molecule. And so we decided we would take the option of putting out on the molecule something that bound to albumin. And why albumin? Well, albumin is there's lots of drugs that bind albumin. In fact, a lot of the function of albumin is to carry things around your body that otherwise wouldn't be dissolved in the water component. So fatty acids, which you need, are all transported through binding to little pencil case type structures in your albumin and get delivered to your heart and to your muscles, where they otherwise wouldn't be able to get there. Mm-hmm. So if you can basically surf or hitchhike on albumin and know how well you can stay there, you can actually, and fine tune that structure. You can basically change the presence, the amount of time that molecule is in the blood. Why is that important? Typically, tumors are poorly perfused, right? So if you were able to look at a whole body PET scan of blood flow, you'd see the brain light up, the heart light up, the kidneys light up, maybe the pancreas, a couple other organs. Other than that, there's not, they take the bulk of everything, right? In a tumor, you see very little typically, unless it's vascular, which is a different issue. It could be a lot of blood vessels. Right. But blood flow, the cardiac output, is an important piece. And so if you have low percent of cardiac output going to a tumor and your plasma clearance is rapid, you're not going to get a lot into the tumor. So can we play with that curve? Can we bend that curve so that you can actually get more of the drug that's staying in circulation, so you have more of a chance through multiple passes to actually get more of the drug you injected to the tumor? That's when I called Jack. I said, Jack, I think we came up with something that would really be well served by doing some mathematical modeling. And I knew Jack's background in mathematics and all the work he had been doing at Invicro with modeling pharmacokinetics, and say, we can actually fine tune different components of the molecule for the affinity for the target, for the affinity of albumin, and play this Goldilocks game of getting it right, so that you get maximal amount in the tumor compared to normal tissue, that you maximize that ratio. And that's the key piece to, hence the name of the company's ratio, which also is Latin for rationale. So there's a real rationale for what we do, and it's important that we get the right ratio. So I'll throw it back to you.
No, that's awesome, and I appreciate you taking us into a little of the history there as well. Just a side note, my father back in the '80s was when he was prostate cancer, the therapy at the time was strontium. I forget what the number was on the thing, but it was pretty much just radionuclide in. I there wasn't all this.
That was for bone pain, and we know the people that developed it. We know them well.
Yeah. So it would come a long way. Although we think of the radiopharmaceutical space, all right, it has sort of let's say a structured genesis back in the '70s, and then it's come a long way since then. You had in that intro, you were talking a lot about sort of the triumvirate that you're working on at Ratio, namely the nucleotide, the cool pharmacokinetic thing that you're doing, and then the linker technology that makes the drug complete. Love to start off with talking about the radionucleotide and what actinium 225 is all about, because as we were chatting before the show, we talked about lutetium previously, understanding that's more of a beta focused emitter. Strontium, I forget what that was all about, but here we're dealing with actinium, with a very rare substance. Read that we create about a grain of sand worth on the planet every year, so kind of a rare substance, yes, but a very potent alpha emitter. Talk about a little bit about actinium 225 and what exactly it means in terms of why we want to bias towards alpha decay in this particular case.
John, you want?
Yes, I mean, we'll ham and egg this to death.
Yeah,
I'm sure.
We
We can both.
have a lot of opinions of all these isotopes, and I think it'd be good to.
Yeah, so actinium is a very large metal, and when you look at radioactivity in general, the bigger the bigger the atom tends to be, the more unstable it tends to be, right? And so for it to become stable, it has to give off lots of things, and so this happens to give off an alpha particle. An alpha particle is a helium nucleus, essentially. It's two protons, two neutrons. It's a big particle for a atom to eject out of itself, right, in order to create that stability that's seeking. Unlike, say, lutetium, which gives off, it doesn't like the electron configuration, so it basically is shuttling things around, and it'll pick out an electron. And that is, that could be a very damaging particle, but it's nowhere near as damaging as something that 8,000 times roughly the mass, right? So alpha particles are particularly damaging over a very short period, a very short space volume, because they have such a big mass and they tear things up as they pass through, right? So they ionize things, they break chemical bonds. Electrons or beta particles can do the same, but they don't do it as well. And photons, which you typically use for external beam radiotherapy, have to give it in copious quantities in order to have the same effect to kill tissue. So this is a very, very potent, alpha particle is very potent, damaging or ionizing particle that can damage things with only a few emissions through a cell. One of the things about that is, because it's such a big and clunky isotope, is that you really need to put that onto a molecule in a very stable way. Now, actinium decays. The first decay happens within 10 days. It's a half-life of 10 days. Basically goes to francium. That has a 5-minute half-life, kicks off another alpha particle. That goes to astatine, that kicks off in millisecond half-life, that kicks off another particle. Eventually goes to bismuth, that kicks off another particle. So every atom of actinium winds up hitting through a cascade, throws out 4 alpha particles, right? So very, very potent, dense collection of large particles to break up the chemical composition of a cell. And that's how these things start to die. And one of the things about it, and I'll throw it back to Jack for more of the physics side, we have a very unique chelator, which is more or less a molecular basket that metals are, the only way to get metals to attach to things is typically to chelate them. That means that you actually put them in a cage where they don't have the same kind of carbon-carbon interact, like a bond that carbon and carbon would have, or fluorine and carbon would have. They need to be grabbed in multiple spots to stabilize them and make sure they go along with the luggage, so to speak. So we have a molecule called macropa or macropod, depending on where you're from. Yep. And I don't know yet where anyone's from, but lots of people say both. And that will actually rapidly bind actinium and keep it on the molecule for weeks. So it's another toolkit Ratio has to actually think about developing actinium label compounds. And I'll let Jack jump in.
Yeah, I'll comment on mass and energy and how it pertains to biology here, which what's exciting about this space is how interdisciplinary the space is. We're a small company at 45 people, and we got 25 PhDs of 7 different backgrounds, right? Nice. It takes a lot of different thinkers to come together. And to kind of recap on the 3 types of radioactivity, gammas, which are used for imaging, positrons annihilate electrons. You get 2 511 keV gamma rays in a PET scanner because E equals MC squared, which is, it's always exciting to use that equation. And you need a lot of gamma rays to impart a really very NIST traceable, measurable amount of energy to a tumor. It's a very regulated space, as more than half of patients with solid tumors get external beam radiation. Yep. Radioactive iodine was FDA approved in 1951 for the treatment of thyroid cancer, still the standard of care. And there you inject radioactive iodine 131, which emits a beta and basically annihilates your thyroid. It's not just used in cancer; it can be used in hypothyroidism and other diseases of the thyroid. And to John's point, the alpha emitters are really the first alpha emitter, and only an alpha emitter approved is just radium chloride called Xofigo, sold by Bayer, used for metastatic bone cancer. analogous to what you were discussing earlier. And to put things in context, if a beta was a baseball, an alpha emitter is a 140 kilogram wrecking ball. And you can calculate this energy deposition for all these particles. And every isotope, to John's point, has different, there's those three particles, but they'll have different energies, and that has different calculations. But it's very interesting. 2 points to take away. The first is when you do all these calculations, if you all we care about joules per kilogram, called the gray, energy per mass. That's by and large the efficacy goes and response goes like energy per mass. And in external beam, they literally calculate what's the energy per mass they're imparting. When you do everything equal by energy per mass, alpha emitters are still on average 5 times more efficacious. So there's something more than energy per mass. To John's point, there's the type of radiation, the biological effect of having a wrecking ball as compared to a baseball going through the DNA, for example, or just the cell membrane or whatever the biology is. So it's an exciting time as a lot of uncalculated things there. We can make the energy calculation, but the biological calculations have to come with data over time, empirically observed. And we're in that space now of starting to see great promise of all these, of betas and alphas. And the last comment I'll make is how you alluded to it with there's just a grain of sand, so to speak, on the planet Earth. I mean, the masses of what we work with are so comically low. Right. And I think that, I'll just take it to FDG PET, which I hope you're not familiar with, but most people will come across it. That's a standard of care image in most solid tumor cancers. It's just radiolabeled sugar. And in that case, they'll inject 10 to 30 micrograms of radiolabeled sugar. To put that in perspective, if you were to take a hammer to an ibuprofen on your counter, you can't really even see that mass. It's so small. And you inject that, and within minutes, it's completely uniformly distributed through the body where sugar would go. So it's amazing what's happening here. And we give drugs at such low mass dose in this space that we have to John's point, there's no pharmacology here. There's no mechanism of action of hitting a target that has a downstream effect. It's how much radionuclide gets there for how long, at what ratio is the most predictive of efficacy. And we operate in very, very unique very unique class of drug. It takes a while to wrap your head around it. But it's exciting, and you can image, and you can always image. To John's point, you can image the patient first. So it is truly precision medicine. I image this patient is going to need. They have the target. This is the right drug to give to that patient.
No, I always love the concept in the sense that, I love the world of precision medicine, but again, precision medicine with the wrecking ball is even cooler, right? I mean, so just, so to recap so far, we have the wrecking ball, which is the actinium-225. We got the trillium, which is doing the fancy pharmacokinetics with the albumin. We got the Maricopa, which is allowing us to now to connect this to all different types of delivery vehicles, antibodies, other agents. So this all forms the basis for ratio. Before we get into some of the deals you've been doing and the clinical work, just say a couple more words about the company, because I know you came out of stealth in the middle of 2022. You raised a nice amount, close to $100 million of funding. Could you walk through a little bit of sort of what the day-to-day is like in Radiotherapeutics, in the sense, I guess most biotech companies that we profile, with I guess more traditional therapeutics, we got our assays, and we got our pharmaceutical work over here, and everything else that happens in drug development 101.
Sure.
What typically happens in radionucleotide 101 land in the lab on a daily basis? And some of the tools that you need, obviously, you don't have a lot of it, but nonetheless, it's sitting around. Where do you keep the actinium-225? So some of these little interesting anecdotes for us. I think that'd be kind of cool.
Yeah. So Jack, let me start.
Yeah, go ahead, Jack.
So in saying there's no pharmacology involved is really when that's in regards to the efficacy, right?
Sure.
Yes.
But there is pharmacology involved if you consider it that, or biochemistry involved
Right.
in trying to understand what makes a tumor a particular cancer, say not even a tumor, but a particular cancer, a potential candidate for radiotherapy, a targeted radiotherapy. And so you have to look at in the case of some of the things I explained, it may not be obvious to you or the audience, but putting a metal on something and adding a big chelate to it, and then attaching it to a smaller molecule, whether it's a peptide or a small molecule per se, organic molecule. You wind up with a construct that is not going to cross membranes without some active transport. So
Okay.
you're making drugs that need to initially find a target on the surface of the cancer cell that is attractive for this kind of generating a ratio that we want, right? So you look at it that way, you're going to be looking at target identification, right? So we have a big biology group, we have a big med chem group, we've got a radiochemistry group, we got a physics group, we got a quantitative pharmacology group. So when you look at this, and we can give you a couple of examples. We did a deal with Bayer on a PSMA assay. So PSMA assay known at the time. What was unique about what we did was that we actually used our Trillium construct as, say with the albumin binder and so it was all fine tuned to give what we thought was the ideal ratio for using alphas, right? That took a lot of time and effort, and then we approached another target called FAP, right? Fibroblast activating protein alpha is expressed on the stroma of most epithelial carcinomas and some other cancers such as sarcoma, esophageal carcinoma, and mesothelioma. That is also a well-known protein that has enzymatic activity. So, like many drugs in the world, they're enzyme inhibitors. So you make an enzyme inhibitor that has strong potency to that enzyme, and then you start to build your constructs, right? So there'll be, again, med chem, there'll be biology, pharmacology, all working on a daily basis to first identify the target, come up with structural leads for that target, generate a significant library of compounds, work with that library of compounds, then gets transferred over essentially to radiochemistry, where they have the expertise of putting various isotopes. So some of the isotopes we can get using a generator that stays in house, short-lived gallium-68. Other ones we have to bring in on a weekly basis or bi-weekly basis, so we have to buy them from bigger manufacturers that make, really some of them don't do anything but make radioisotopes, right? So you can buy actinium from the Department of Energy, which is part of the U.S. government. You can buy it from people abroad, and there's a couple of other companies now starting to make actinium in North America. Lutetium, same. There's other companies around the globe that will make lutetium that you can purchase. So, we have a significant effort in med chem. We supplement that with external. Chemistry is complicated and expensive, so you have to supplement some of that with external input from CDMOs. Biology is pretty much taken care of all in house. When we have biological questions, so we don't want to bring every technology into the house, or it becomes expensive. You only use it once a month, so we'll send out things to be assayed by people that have particular expertise. The radiochemistry gets done in-house. We do all the animal work, all the cell culture work, all the enzymatic assays all in-house. And then when we start to develop the actual data in animals, then obviously the physics group and the quantitative pharmacology group get more and more involved on the assessment and the analysis of that. So I'll hand that over to Jack, but that's kind of a day in the life of Rishi.
As a numbers person, I'll briefly just follow with concrete numbers. We have in our database, we have a very good data model here where all from chemistry through that whole process is all captured in our database, and we have designed over 400 molecules for the few programs we worked on. We've synthesized almost all of those, and we've done well over 200 radiosyntheses of actually doing the radiochemistry and radio labeling them. You don't radio label every drug. Sometimes you will kill a drug in an in vitro assay first for some reason.
Right.
Well over 1,000 in vitro assays we've collected and processed over 20,000 biological samples through the founding. And then we were just 3 years old as a business. So from a
Jack, just to be clear, we've only had the lab open for about 14 months.
Right.
Yeah. So we were outsourcing some of that prior, so we did have some data in, but that has ramped up dramatically. We've really hit our stride in this. We're in a 19,000 square foot facility here, about half lab, half office in the Seaport District of Boston. And I think John and I have a lot of overlap in our knowledge base, but also are in and of ourselves interdisciplinary, and then an interdisciplinary leadership team. And you're almost only as good as your weakest link in our having. Both of us have been in this space doing these kinds of studies for a long time. And I think we've built a team here that has, you always try to hire smarter than yourself and brought in experts across that full spectrum. So the only real difference is, there's other differences, but we can talk about going into the clinic here is very unique, with great collaborators in Germany and Austria, in what's called compassionate use there. And so staging the data, doing the relevant toxicology to move to a partnership where people are going to try to treat patients, image, then treat patients in compassionate use, which is akin to how Novartis's Pluvicto and Lutathera for prostate and neuroendocrine, respectively, were started as well. And John was a pioneer in that on the PSMA front. He already alluded to his work with Uwe Haberkorn and Heidelberg. So it's a very interesting class of drug. It's easier to go, no go in this space because you can kill a drug right out the gate. If it just doesn't have the imaging profile of, it's not going to work. You can't image it well. If it doesn't have the right ratio in an image, it's not going to have efficacy. Right. And that's a huge differentiator from people who can take a half a decade to a decade to really understand a clinical efficacy signal just by the nature of their platform. Thankfully, they do that because we have a lot of great drugs as a result. Yes. This is just sometimes maybe an accelerated.
And I think, Jack, to your point, I mean, the imaging allows you to see problems as well, right? So you unlike a lot of other drugs, that are particularly useful, that might have a very narrow therapeutic index, right? Because you don't know a priori that, say, cisplatin is going to the kidney, or adriamycin is affecting the heart. I mean, there's things you don't see in a conventional drug, which is, to Jack's earlier comment when he introduced himself, Big Pharma was starting to get interested in a job I took at Mass General. Big Pharma wanted to know where their antibiotics went, right? So they had no idea. And in fact, and then you had to correlate. Well, you can't image everybody. So what's the correlate between how much is activity, what's your concentration in the blood, what's your concentration in the brain, what's your concentration in another organ? And it's typically not possible to do all that to get that information for conventional chemotherapeutics and other drugs. So we actually start there. We start with the imaging. We start with the ability to see where the drug goes in the mouse. We see, hey, we've got the right ratios. We then make some, I would say it's fairly small steps compared to a big trial to actually get first-in-man data, which allows us to confirm by the image alone, are we on track? Is this really still going to be as good as we think it's going to be? And then from there, rapidly accelerate, right? Because if you saw it, one of the things that's nice about this is you can do very early kill studies, right? Doesn't work in the mouse, it's out. If it doesn't work in man after it worked in the mouse, you're out, right? You're not wandering around saying, oh, what's the right dose and what's this happening and some other thing that, or maybe we can use it on some esoteric application. You would know that you can't do therapy because, let's say, half of it goes to the kidney and it doesn't wash out of the kidney, right? These are non-negotiable things that you can see in radiopharmaceutical.
Yeah, that's elegant, especially in this era of companion diagnostics being developed after the fact, and in the sense you're developing them in parallel, well, even beforehand to so it's a double whammy there, which is awesome. Talk a little bit about, clearly, as you both mentioned, this is really a novel therapeutic class, but you've been generating a lot of excitement, as I mentioned, close to $100 million of funding. You already have partnerships with the Bayer and Merck and Lantheus. And you're developing, I think the lead candidate is something called RTX 1363S, which is you're developing both as a radio pharmaceutical for imaging and therapeutics for something called fibroblast activation protein as a target, which is you could tell us a little bit about where we find that, but tell us a little bit about the plans for RTX 1363S on both fronts, and then I know that sort of copper 64 comes in here. Walk us through that as well, and a little bit of sort of the little intricate package you've created with this one.
Yeah, John, I'll talk about the programs and you talk about FAP, and then we'll. Yeah, just to clarify, in most drugs, it's often frowned upon for a company our size, or almost any size, to say I'm going to develop a diagnostic and a therapeutic. If you're Roche, Roche is pretty unique, then you're in both spaces, and as are some other multinational massive companies.
Sure.
In this class of drug, you really have to develop a PET agent. It is required to have a PET agent to do the imaging first. And in this case, 1363S, which you refer to, we developed and out-licensed to Lantheus, a very successful publicly traded company in the field of PET diagnostics. They own and sell Pylarify, which is really the gatekeeper, doing incredibly well in the PSMA prostate cancer space. And crucial in that landscape. So in this case, we developed it. We worked with our collaborators in Germany and tested some, they were using it as a diagnostic. That molecule is not conducive to being a therapeutic. And so it doesn't have the right residence time and thus not the right ratio for the calculation of absorbed dose that we talked about earlier. And so we have a different class of molecule, and that's what we're marching forward and have worked with our collaborators and are marching forward to filing an IND in, we're targeting a Q4 IND for opening up trials in the United States. And so, and then John, with that, why don't you talk, FAP is a very exciting target, maybe talk through FAP and why it's such a relevant target.
Yeah, so FAP is very interesting for lots of different reasons. And one of them, so first of all, it's a prolipeptidase, so it has enzymatic activity where it recognizes proteins in peptide sequences and will clip them off, right? So it has this very active way of chopping up peptides. It's typically found on activated fibroblasts. So if you before we even get into cancer, so if someone has an injury, whether it's a cut, right, or you think about it, if you're talking about cuts, is probably the simplest way to think about it, right? So you cut yourself, and then you have or a gash. The first thing that happens is that you get platelets. First of all, you're bleeding, then you're delivering platelets, and you're delivering lots of other things to stitch up the bleeding, right? So you think about it from a wound healing perspective. Once the bleeding stops and you scab over, the body then has to figure out, how do I stitch that tissue back together, right? So in a typical wound like that, the body would send out signals, or where the cut is, to recruit fibroblasts to come in to that situation and actually stitch that tissue together. So, which was, some people have different degrees of keloid formation. Like you have a scar, some people would scar very, I would say very minor, like if you cut yourself. Other people, you might see a raised scar and a lump, and they have different amounts of fibroblasts that come into that space, right? So we're all different. And so there's this recruitment of fibroblasts. They have elasticity, they stretch, they basically knit your tissues back together. And so they're typically only present during an injury or a wound to heal the wound. Okay. Now you can actually wind up with diseases and other things where, you have long-term problems with the shoulder and arthritis because it's a constant irritation and a constant wound. You constantly get fibroblasts coming in. Then you wind up with fibrosis, right? You have all these fibroblasts, and they're starting to come in and do things, but they can't really fix anything, and they wind up with so much fibrosis in, say, a joint, that you lose range of motion. So there's and it happens in other tissues, the lungs and heart and tissues. So this is a natural way of healing the tissue. However, when you have a tumor has been described historically by some as a wound that will not heal, right?
Right.
And so the same signaling goes out is, hey, listen, tumors are very clever at co-opting your body, right? You're shutting off your immune system so they can grow and getting more vessels so they can grow, bringing in stroma, right? Meaning, sort of the fibroblasts so they can support the structure. So you wind up getting signals from the tumor and fibroblasts go in there. So it turns out that in most epithelial carcinomas, lung, colon, breast, you have this big recruitment of activated fibroblasts that they're just recruited, and they're sort of been kidnapped by the tumor, and now they're present in a significant portion of the mass of that tumor. So the tumor is a swelling, right? And so you have cancer cells in the tumor, so to speak, and you have fibroblasts in the tumor, and it's like a bird's nest, right? You have the eggs being the cancer cells and the nest being the host and all the tissues. And so what you wind up with is having a really interesting opportunity to go after that fibroblast cluster in a tumor. All right, so it becomes very interesting. Turns out that sometimes those tumors actually have on their surface that same fibroblast activating protein alpha. It's actually on the tumor. Most of the time, it's in the stroma. It's in the cells adjacent to the tumor cells. But in sarcoma, esophageal carcinoma, and mesothelioma, it's also on the tumor cells. Now we have the ability to hit the tumor cell and hit the stroma. So again, to go back to the deal we did with Lantheus, we sold them and developed and sold them and delivered to them what we think is best-in-class FAP imaging agent for PET scanning. And then we had to go back to Jack's point. That wasn't significant retention. So part of the therapeutic index is not just that you get good uptake, but that uptake is prolonged because you need to have residence at the tumor site in order for your isotope to decay, right? So if it went in there and all you saw was the tumor and an hour later everything was gone, you didn't deliver any activity, right? You think about it as cooking. You have to bring the oven up to temperature, and then you have to cook for a period of time. Because if you put a turkey in a 500-degree oven for a minute, nothing's going to get cooked. Right. You put it in there for 5 hours, it'll come out as charcoal, right? So there's a temporal component to this. You get the selectivity and the ratio, but then you need this time element. So we went back to the drawing board after we delivered the compound for Lantheus and tried to design into the molecule a way of having it stay where we want it to go in order for us to get up to temperature and stay at temperature, right? For us to deliver the radioactivity on injection, but let it sit there. And so that's where we are now. So 1363S is the diagnostic drug that we licensed to Lantheus, which is now in their hands for the clinical development. And we've got another compound, 2358, that we're looking to put into the clinic as a therapeutic. It has a different structure than the other one. It goes after the same target, but it's very different kinetics. And so this is now the evolution of our first non-deal therapeutic that we'll bring into the clinic ourselves.
Excellent. Really?
And that'll be in sarcoma. That'll be in soft tissue sarcoma. Got it.
And just if I forgive me if this is a silly question, but clearly the primary use of radiation-based therapies throughout history has been in the oncology space. As you develop these innovative ways to administer these unique isotopes, are there non-cancer possibilities here? You think things like how a lot of cancer drugs are repurposed for autoimmune diseases to slow down growth of non-cancerous tissues. Any interesting possibilities there, or is it just a little too early in the state of things to go in that direction?
I think there's always the possibility. I think it is early. I think one of the things you always have to worry about with giving radiation, no matter what that radiation is the potential of causing another cancer, right?
Sure.
It's just like chemotherapy. There's always a price to pay when you have something that's going to kill something or change its DNA in the process of killing it. You have to be concerned about it. Right? It's like any other drug. I mean, even drugs that aren't cancer drugs might have an increased cancer risk with them. We just don't know until you have enough exposure. But the idea that there might be a potential, I think, is real, particularly for FAP, where you have really debilitating fibrosis affects a lot of people, and there's some other things that have to do maybe
Sure.
with the immunological response. So, Jack, I know you had some of your own thoughts on this. I think it's
I think it is early days, and it might not be an isotope like actinium, which it could be that a beta emitter. People still do use external beam radiation for debilitating arthritis, for example, in really more elderly patients where the cost benefit and the benefit analysis of the risk of getting cancer from the radiation is greatly outweighed by the benefit of crippling arthritis in a septa or octogenarian, for example. So we're very curious. We have good cause to monitor and see if there's going to be impact on arthritis, for example. Here, I just think we've covered kind of the range of the side effect cost benefit analysis. So it's going to have to be something like what was discussed. Right. I would say, and radiation is used for hypothyroidism. It's not only used currently. Right. Yep. I did want to add, because I think we've been talking a lot. It's so interesting, this class of drug, you really measure the toxicity, to John's point. We can image the toxicity. We know where it's going to be toxic. But I want to put things in perspective in your nicely named podcast. We've talked a lot about progress. We've touched on potential, and when we think about, and we just discussed possibilities a bit. I will say the great hope of this class of drug is combination therapies and a replacement for chemotherapy. We've talked a lot about the side effects here, but truth be told, when people take Pluvicto and they have metastatic prostate cancer, they'll ask if they're on the placebo. They'll say, I just feel very normal. I feel great. Like, I'm not feeling side effects. It can knock down your platelets a bit. It certainly will over a long period of time have a longer period of time could induce chronic kidney disease. But when we compare that to certain current cancer treatments, like right now, we're giving a lot of drugs in combination with immunotherapeutics while simultaneously destroying people's immune system. And so I think that the hope here is that not just as single agent therapies, which is where everything has to start and where we're starting this fall, and have started with our collaborators already. I think there's a real hope here that for solid tumors, when you combine these drugs, which generally do not weaken the patient per se, the mantra of our business is extend life, maintain quality, that the quality of life after these drugs and while on them, it's very high. We're just really hopeful around the possibility of and that's why there's been so much excitement for this class of drug. That's part of it, is a replacement for what are really tough therapeutics. I hope you haven't witnessed firsthand, but I know John and I have. People live through chemotherapy, and it's tough. It doesn't mean that it doesn't have a place, and in certain indications, it's quite effective. But in many cases, it's not. And this is kind of the great hope of this class of drug.
I can vouch for lutetium, because I mentioned my dad with the strontium a couple decades ago, but my father-in-law was on Pluvicto, and I can vouch for. I mean, there wasn't a side effect to be seen. He was happy. The only side effect was he couldn't be near his wife for a couple of days. But no, I saw it firsthand. So I think the potential here is amazing. We've come a long way since the 70s, and that's extremely exciting. Can you guys say a couple words? I mean, I know you recently, and John, you mentioned something about it early on in terms of the manufacturing. You did this deal, I think, a couple of months ago with PharmaLogic to start to help to scale up this first therapeutic. Can you say a couple words about sort of what's involved in that? I mean, how much beyond are you making a grain? Are you making 2 grains? And have there been any sort of general Leslie Grove type moments with, don't drop that plutonium, it cost $50 million, that have happened along the way?
I would say, actually, I'll quote John. He didn't chime in. John says it best: you're shipping melting ice cream unrefrigerated. It's decaying. It's so unique as a class of drug. Every batch of drug has its own release criteria. So good manufacturing practices are on a per batch basis, unlike having lotted, stockpiled drug product. And we have a great partnership with PharmaLogic, also a good relationship with Eckert Ziegler in Europe. And we're right in the middle of our first clinical GMP campaign for this trial. And I think long term, I would forecast that there's a lot of commentary about isotope supply now. I think the trains left the station with governments and large scale enterprise putting near $1 billion into the long term supply. John and I have been in this space for a while. If there is need for radioisotope, we as a species are going to be able to produce it. And so I think we're going to see this landscape change, but our long-term prognosis is very positive on manufacturing and logistics. And Novartis, thankfully, the big players are here, and Novartis has done a great job, in my opinion. They've taken some heat, but openly, they've, in my opinion, done a fantastic job there's the demand greatly outweighed the supply. And so that's going to catch up. Where there's a will, there's a way. And where there's a will and economics, there's a way, would be my
suggestion. And I'll just add that, the agreement we have with PharmaLogic is key, right? Because we have, you can't be good at everything. And the manufacturing of a radioactive drug has really nothing to do with discovery, right? It has nothing to do with all the things we talked about and what we do on a daily basis, right? It has a very strict set of rules, similar to any other GMP manufacturing, with the complexity that you have a radioactive drug. So typically you're protecting the drug from the operator, right? And you make sure it's sterile and it's in the right environment. But here you have to protect both. So even the physical plant's complex in terms of how do I make sure everything's sterile and all the air handling and everything is correct, and the operator, the person that's actually has to run the system, is capable of being protected as well. So PharmaLogic has thought this through in super detail, right? And they have a great track record both on their diagnostic manufacturing and their therapeutic manufacturing. So it was really important for us to get an agreement with PharmaLogic, also Eckert & Ziegler, which if you go back, we have a good relationship with them as well. These people have spent their life not in the discovery and clinical trial piece, but in the manufacturing piece, and you need that, and the field needs that. And to Jack's point, the isotope manufacturing, production, whatever you want to call it, depending on how they get their sources, that's another piece. So it's separate from us. It's separate from the PharmaLogics of the world. And that's also important, right? These are people that are running nuclear reactors or accelerators that have the sort of nuclear chemistry capabilities of making isotopes at the purity they need, and then shipping them to CDMO, who then we contract with to make our drug. So there's a real vertical chain there that's really important, and for all of us to be successful in the field. And I think, to Jack's point, Novartis jumping in, and I think they've probably taken over a lot of, I would say, primitive, not in a negative way, but early stage manufacturing. Now they're bringing big pharma manufacturing thinking to this space, and others are as well. So I think it's really crucial for us.
Yeah, PharmaLogic, to John's echo, it's a very regulated space, very logistics heavy demand. They're a phenomenal company, and we're seeing, Ira, across the space. Let's be open. A lot of money and resources have come into the space over the last 8 years, and we're seeing there's going to be evolution of everything, evolution of regulations, evolution of partnerships, evolution of integration, evolution of adoption. But in the end, people are seeing great efficacy and would prefer. That's what's going to drive. Everything else will follow if you have that.
Absolutely. Guys, anything I didn't touch on that you want to mention, either related to the clinical programs, other assets in the portfolio that are coming online, anything I missed, conferences that are coming up, or talks you guys will be giving, please both of you take the floor with final messages.
My quick messaging would be, big nuclear medicine meeting coming up in Toronto. It's a Society of Nuclear Medicine. We will be there. We've got a couple of presentations. These meetings, as you can imagine, over the last 5 years have been getting more and more buzzy. There are lots of excitement, lots of new companies. There's companies we haven't heard from before. Very exciting time. When it comes to our own pipeline, we've got 4 targets we're working on behind FAP. We're looking at exploiting the Trillium platform and the Actinium platform and the Chelate platform to really make best in class on all these targets. So I'll just leave that and hand it back to Jack.
Yeah, no, I think everything's been said well. I'll just close by saying, I think, not to use a baseball analogy, but I'm going to, we're in the second or third inning here of this sport. And, there's a lot of promise and a lot of things we said today were very predictive, and maybe we're going to change course on certain items as we get more information, because data drives decisions. But there's some core principles here that are looking very good, and we're very excited about the outlook and excited to be part of what is a big push in society to fight cancer, to extend life and maintain quality. I think that says it best.
Yeah, I'm excited for you guys and rooting you on, and just happy to see you guys taking I call them risks, but I mean they're risks. I mean,
there's plenty of risks.
Yeah, I mean there's risks, but at the end of the day, these are common sense approaches to an area that
worth, Ira. They're well
exactly.
worth
Calculated risks.
Yeah, extremely calculated. Both of you guys clearly know where you're going with this. And again, I really enjoyed the discussion. I'll look forward to continue to follow you guys and maybe do a follow-up next year to see where everything is. But really great stuff. Again, for everybody that are going to be listening to this episode of our show across the various podcast networks or watching on our YouTube channel. Again, you've been spending time with Dr. Jack Hoppin, Chairman and Chief Executive Officer, and Dr. John Babich, Director, President, and Chief Scientific Officer, Radiotherapeutics, doing really amazing work to move forward these best-in-class targeted radiotherapeutics for the treatment of all cancers. Jack, John, I really want to thank you both. It was really great spending time with you, learning about what you're up to. Obviously, thank you for everything you're up to. And as we like to say here on our show, thanks for helping to create a better tomorrow for millions of patients out there. Really a great story.
Thank you.
Thank you, Ira. Real pleasure.
Thank you for your time.